Chapter 19. We've only got 3 chapters left, including this one, so we are nearing the end. So this one is going to be a short and fast one, taxonomy, systematics, and phylogeny. This one's a pretty interesting one. It's looking at the systems of classification and how organisms get classified. And I know we kind of touched on this previously, talking about kingdom, phylum, class, order, family, genus, species, and how domain got added in there as well. So we're just going to expand on that in this chapter. So the systematic of biology, the 3-domain system, and then phylogeny. Phylogeny is looking at that classification and how everything is related and looking at kind of like a branching family tree history and common ancestors and how everything branches off of everything else. So taxonomy is the branch of biology that identifies, names, and organizes biodiversity into related categories. It's kind of neat how everything is related, and we already talked about how there's like homologous and analogous structures, embryological development that looks similar, how there's convergent and divergent evolution, and geographic isolation, and all of these different things in the fossil record that makes organisms appear to be similar, and how there's evidence that points toward a similar common ancestor. So the naming and identifying organisms began with the Greeks and the Romans, and all organisms have a Latin name because the Latin names were created that Latin names don't change. At least that was the idea, was that by using Latin words, this was an ancient language that doesn't change very often, so that their naming structure wouldn't change over time. Aristotle classified organisms into groups such as horses, birds, and oaks, so kind of the bigger classification. In the Middle Ages, organisms were described using the Latin names, and so that's what's kind of stuck around, and these Latin names are still used today. Now, there's a lot of advantages to using a common universal classification, because what I call a bluebird here may not be the same thing as what somebody else calls a bluebird over in Great Britain or Australia. So having a universal name for a bluebird just helps to unify and clarify what a bluebird actually is, so that if I say Rana cataspiana, we'll know that that is the bullfrog no matter where you're at. So it just kind of helps to clear up any confusion about what the common name is. So the common name would be bullfrog, but the scientific name is Rana cataspiana, which is the Latin name. So here are just some examples of different types of organisms, and everything has a common name and a scientific name. So we are humans, and that's our common name, but our scientific name is Homo sapien. So again, just kind of clarifying that and making that distinction. Vertebrate animals are where we have the most trouble because there's a lot of different names specifically for this one. This one has a lot. You could know it as a mountain lion. You can know it as a cougar. You can know it as a puma. So this one, a catamount, is another. This one has a lot of different names. But Felix concolor is the scientific name for it, and that kind of helps to clarify and clear up any confusion that it might have. So in the mid-18th century, Carolus Linnaeus is the one that really kind of helped to clear this up, and he came up with this idea of binomial nomenclature. So binomial for two and nomenclature for name, and his idea was the genus and the species. So that is the most basic. So genus is like second on the rung, and then species is the most specific. So if we are Homo sapiens, Homo is our genus and sapien is our species, and that is our scientific name. You put the two together and that gives us our scientific name. Now if you look here, this is the correct way to write a scientific name, which is really important. We have our genus here, Lilium, and we have our species over here, Bilbiferum. So Lilium is the genus, Bilbiferum is the species. The genus is always capitalized and the species is always lowercase. The entire word is italicized. Now if you're writing this on paper, then it would be underlined because you can't really, you could try to italicize it on paper, but it's easier to just underline it. So when you're typing it out, the entire word is italicized, but if you're writing it out, the entire thing would be underlined. So the correct way to write a scientific name for the binomial nomenclature, genus is capital, the rest is all lowercase, and then if you're typing it, it's italicized. If you're writing it, it's all underlined. So all binomial nomenclature, every scientific name, it's based on genus and species. So you can usually tell just at a glance if organisms are related, because if they have the same genus, that means they're closely related. So Lilium bulbiferum and Lilium canadensis, they're sharing the same genus, and so that means that they are very similarly related because they have the same genus. So they are only differing in the species, canadensis and Bilbiferum, and so that means that yes, they are different species, but they have a very close ancestor because they share a genus. So it helps to, at a glance, tell what they are and how closely related they are. So if they don't share a genus, then that means they are more distantly related. So here's a picture of Carolus Linnaeus. He was really good at classifying things. Here are the two flowers we were talking about, the two lilies, Lilium canadensis and Lilium bulbiferum, and you can see that they do look they look very similar, but there are a lot of differences as well. Coloration, the way that they're hanging on their stems, just the overall structure. So Lilium canadensis, Lilium bulbiferum, they are kind of like cousins, you would say, but they do have a lot of differences as well, so they are not the same species. Now here is the order of classification. I know you've heard me say it before. This is what is currently used. Now with Carolus Linnaeus, he did kingdom through species, and domain was later added on. So there are 3 domains and 5 main kingdoms. So kingdom, phylum, class, order, family, genus, species, that was all made by Carolus, and then domain was added on later, and there are 3 main domains. So there are several acronyms or sayings that can help you remember this, and one of the more common ones is definitely keep pond clean or froggy gets sick. So you can make up many different kinds to help you remember this, but definitely keep pond clean or froggy gets sick is a really good one to help you remember the order. Eventually you'll be able to just spout it off if you practice it long enough, but for now using an acronym would be very helpful. So this is the most specific and this is the most broad, all-encompassing. So this is just some examples looking at how they get classified. So mammals versus amphibians. So mammals is in a different group than the amphibians would be. Now, the higher the category, such as domains, the more inclusive, so that means the more broad and the more different things can be classified. So the species is the more exclusive, so the more specific. That's how I remember it. Species is specific. And that means that there are fewer things in it, and only certain types of organisms can be classified as a species. Now, a species itself is an organism that is very similar and can breed and produce fertile offspring. We already mentioned this before. So like a donkey and a horse can breed, but their offspring are sterile when they produce a mule. So that means the donkey and the horse are different species. They are not compatible. And so a dog is a really good example of a species. A dog can breed with any other kind of dog, and they can produce a mutt. And so those mutts can produce other mutts because they are fertile. So they are all the same kind of species. And yes, we have different breeds of dogs, but they're all dogs. They can all interbreed and produce fertile offspring. So kingdom previously in eukarya are now supergroups. So scientists are so weird. There are like two groups of scientists. They're the type that are strictly kingdom, phylum, class, order. And then there's the type that are like supraorder and suborders and superphylums and subphylums and then there's like subspecies and they divide it out more. So it really depends on like which category you're in. Some people, some scientists are really against like subspecies. They're like, well, it's just a species. Why are we going down to subspecies? So the other thing to remember with this classification is it's changing all the time. In science, some things stay the same for a very long time. Classification is not one of those. Classification is changing all the time. There's so much debate, and part of it is now that we can look at the genetic and the DNA level, we're discovering more and more new things. Like a great example is the red panda. The red panda, it's called a panda. It's not a panda. It was grouped with the pandas originally, but then they decided it wasn't a panda. It's not a bear. And then they put it with the raccoon because they thought it looked like a raccoon. Well, and then they decided it wasn't a raccoon. So finally they just put it in its own group because they thought it was just weird enough that it just deserved its own group. So that's where it is right now. So classification is one of those things that is just weird. It just changes all the time. So keep that in mind as you are learning about classification. It's just one of those things that as technology develops, as new animals and new evidence is found, things are going to change. This does not stay the same. And that's why there are super orders and subgroups and supergroups, is because this is going to change a lot. So the task of creating standardized rules and nomenclature is difficult and has most recently been aided by the process of DNA barcoding. Again, new technology creates new complications in this. So it compares short fragments of DNA sequence from an unknown organism to a large database to help group these animals in better groups. And remember, this isn't just animals, this is also plants. Plants are part of this process. So traditionally, taxonomists relied on anatomical data. Does this animal look like this? Does this one have the same length of tail as this one? It works, but it's not exactly accurate. But then we have the Consortium for the Barcode of Life proposes that all scientists will be able to identify a species with the flick of a handheld barcode. That's kind of cool. Like the 11-digit Universal Product Code, the UPC used in supermarkets, the scanners on like the barcode on products. The DNA is the UPC of organisms on Earth. So yeah, organisms that are alive have DNA. If it's alive, it has DNA. That's part of what makes living things. A DNA barcode device would provide a fast and inexpensive way to catalog organisms. That'd be very cool if you could scan something's DNA and figure out what category it belongs in. DNA barcoding has been criticized as being too simplistic, but it is a powerful way to catalog a portion of the world's biodiversity. It would be pretty cool, wouldn't it? So going back to the three main domains, we've got domain bacteria and domain archaeobacteria. So domain bacteria consist of prokaryotic, because again, remember, bacteria are going to be your prokaryotic ones, the ones that do not have a membrane-bound nucleus. And archaeobacteria are also a type of bacteria, but these are a little bit different. They live in extreme environments, and they're going to have a plasma membrane and a cell wall, and the cell wall is going to be a little bit different than the other bacteria, and that's going to be one of the main things that separate them from the domain bacteria, is their cell wall is a little bit different. They're going to also have kind of a little bit of a different RNA the ribosomal RNA, and the nucleotide sequence is going to be just a little bit different as well. So there's some fine-tuning differences between domain bacteria and domain Archaeobacteria. Now, domain Eukarya is the largest group, and this is the one that we are in: protists, plants, fungi, and animals. So a bigger diversity here. And due to difficulty in classifying the protists and other level of classification, the supergroup has been created. So fungi are mostly multicellular, plants are photosynthetic, animals are motile. And so we've got these different groups: protists, plants, fungi, and animals. These are the kingdoms that fall under the eukaryotic domain. So again, we've got domain bacteria, domain Archaeobacteria, and then we have the domain Eukarya. And under the domain Eukarya, we've got protists, fungi, plants, and animals as the kingdoms. So this kind of shows how it branches off. So we have our common ancestor back here, and that gave rise to bacteria, Archaeobacteria. And if you look, Archaeobacteria gave rise to the Eukarya. So they had a common ancestor here. So really the most primitive was the bacteria. And the bacteria gave rise eventually to the Archaeobacteria and the Eukarya. So that's kind of interesting. Now this is a phylogenetic tree. This phylogenetic tree shows relationships. Anything closer to the trunk of the tree is more primitive, and anything out toward the branches of the tree is more evolved and closer to is more advanced. So that's kind of how you read this phylogenetic tree. Anything closer to the trunk is more primitive. Anything out toward the tips of the tree is more advanced and more evolved. So here we have a common ancestor. Anywhere we see kind of something meeting a major branch, that's a common ancestor. That's another one, and that's another one. So here we have a common ancestor that branched off, and here we have another one that branched off. So major distinctions among the three domains of life. So bacteria, Archaeobacteria, and Eubacteria. So bacteria and Archaeobacteria are all going to be unicellular. Eukarya, they are going to have some that are unicellular, especially in the protist group, but many are going to be multicellular too, like us. Oh, excuse me. Membranes, they're going to have phospholipids and varied branched, and then they're going to have phospholipids and bibranched. Cell wall, and this is where there's a little bit of difference. Peptidogolin is found in bacteria, and no peptidogolin is in Archaeobacteria. So that is where one of the differences lie between bacteria and archaea. And, oops, sorry, didn't mean to hit that. Archaeobacteria and bacteria, some and some are not. Some of the Archaeobacteria have a cell wall and some do not. Plants do, others don't. No nuclear membrane and no membrane-bound nuclear organelles, but we have those. And ribosomes and introns. Remember, introns are in the protein-making process, and introns are what are removed during the protein-making process. All right, so moving on to phylogeny. Phylogeny is looking at past records, specifically fossil records, and how they have changed over time, and looking at that phylogenetic tree and how organisms started off with a common ancestor near the trunk of the tree and how that common ancestor has changed over time and evolved over time. So ancestral traits present in all members of a group. Sometimes those traits can be lost though. So at one point they had them, but the traits can be lost over time as well. So it could have started off with a specific trait, but it can be lost over time. So derived traits are present in some organisms, but absent in a common ancestor. So maybe it evolved later, which is why it makes phylogeny so confusing. It's present. It could be an ancestral trait, or it could be a trait that evolved later on. And an example of a derived trait is an opposable thumb. It's not present in the common ancestors of all mammals. It is an ancestral trait of primates. So ancestral trait is present in all the common ancestors. So it really kind of varies depending on what trait it is we're talking about. But sometimes we can see where that trait came from, such as here is where we predict the even-toed hooves evolved, and then the animals that came after that had even-toed hooves. Here is where we predict the mammary glands developed, and everything that came after this point all had mammary glands. Here is where we think the opposable thumb developed, and everything that came after this point is where the opposable thumb came. And we think that the shoulders came here, and so that allowed anything after this point to develop good shoulder blades and kind of the rotational pattern. And then it can get even more complicated depending on the type of animal, the type of features that animal has, and so on and so forth. So we can kind of work our way backwards depending on what we know, and we can kind of fill in the gaps. And we can use the fossil record to help us fill in those gaps so we can make predictions about how animals are related. So by looking at what we have now, by comparing what we have now, and by looking at the fossil record, we can usually figure out, okay, these animals must be closely related. They must have had an ancestor here, and this is where they got that trait. And by doing that, we've been able to figure out quite a lot about the animals, again, using those fossils as an area to fill in the gaps. Now, cladistics is a method that uses shared derived traits to develop a hypothesis of evolutionary history. So, evolutionary history is derived traits is interpreted into a type of phylogeny called a cladogram. A cladogram is a special type of phylogenetic tree. A clade is an evolutionary branch that includes a common ancestor and all of its descendants. So this will make sense in a little bit when we look at a picture. A cladogram is basically a working hypothesis. And remember, hypotheses can change. And hypotheses is just a prediction. It's not true, it's not false. We're just trying to support it with the best abilities that we have on hand. Hypotheses can change when new traits are discovered and incorporated into the cladogram. So it's changing. It's constantly changing. It's constantly being tested. And as new evidence is found, it can change again, and that's perfectly fine. As I mentioned, classification is something that is constantly changing, and that's normal and that's expected. Now, cladistics is guided by the principle of parsimony. The minimum number of assumptions is most logical. An organism probably didn't go through a very complicated process. It probably took the most simple path. Now it could have gone and through a really complicated process, like for instance, to get our opposable thumb, it could have gained a thumb and then lost a thumb and then gained it and then it did this and this and this and then it finally got the thumb. But that's probably not what happened. It probably got a mutation and got a thumb. I mean, the simplest way is probably what happened. It could have been more complicated, but most likely not. So that's kind of what this cladistics is based on, is we saw this appear here, and that's the assumption that we're making, because anything more complicated than that, nature probably can't handle. So nature's going to take the simplest way and go with that, because nature doesn't really handle complicated well. And so that's what parsimony is talking about, taking the most simple way and then running with that and focusing on that. The reliability of cladograms is dependent on the knowledge and skill of the investigator. So really, there's a lot of interpretation. There's a lot up for interpretation here. It's doing its best to make those assumptions based off of what we see, what we know, what information we have available, and what the fossil record is showing us. So here we can kind of see we've got our species in group and we've got the traits. We know that a chimpanzee has mammary glands, has hair, has an amniotic egg, forelimbs, vertebrae, and notochord and embryo. It does not have scales, it does not have a gizzard, so we didn't check those. And then we can kind of keep going. The only one that has all of this is the, what does that say? The lancelet, which is an outgroup. which means it's a more primitive group. So we can kind of compare and contrast by using a chart like this. And we see that the finch and the crocodile actually has the most similarities, which really isn't surprising because birds and crocodiles are actually fairly closely related. They have a lot of similarities in common. And a dog and a chimpanzee are both mammals, so it's really not too surprising that they have the most in common too. A lizard and a crocodile have a lot in common, and that's not terribly surprising because they're both reptiles. So making a chart like this is really helpful because you can see who shares the most traits. Then we can put all that into a tree, and you can see that here's a common ancestor and so on, and this is where we think each one of these traits have developed. And if that's true, it gives us a fairly accurate table showing that a dog and a chimpanzee are both mammals and that a bird and a crocodile actually are pretty closely related, which is true. There's evidence that shows that the bird branched off of reptiles, specifically a crocodile, and that it was able to develop on its own. There's a lot of indication between these. Crocodile and alligators have four-chambered hearts. Birds have four-chambered hearts. They make a nest, and the birds make a nest. Crocodiles take care of their young. Birds take care of their young. There's a lot of similarities between these two that suggest that they branched off from each other. Homology. Homologous structures are related to each other through common descent. The forelimbs of vertebrates contain the same bones. We already kind of mentioned this before, so I'm not going to spend a whole lot of time on it. Convergent evolution is where you have two types of organisms that develop closely to each other because they have the similar selective pressures on them, even though they live in different places. They may both be in grasslands or they may both be in rainforests, and because they live in similar habitats, they have the same pressures, and they've developed a habitat closer and similar to each other. Analogous means that structures do a similar function, but they are very different, such as the flippers on a dolphin and the fins on a fish, both used for swimming, but they're very different. The flippers on a fish fins on a fish are nothing like the flippers on a dolphin, because the flippers on a dolphin are like hands, and the ones on a fish are like fins that are bony and don't really have hands at all. So here we can see an angiosperm and ancestral one, what it looks like. And then we've also got some behavioral traits which can be taken into account when trying to classify organs or organisms. Parental care, mating call, mating seasons, molecular traits. Two species with similar base pairs are assumed to be closely related. Two species with differing base pairs are assumed to be only distantly related. So looking at the genetics, the closer the genetic history, the more likely it is for an organism to be related. So here again, some other examples. We should see a cow and a pig and a horse. All of these actually are mammals, but we can see that the human and the orangutan should be pretty close, but actually the human and the chimp should be very close because the human and the chimp have very similar DNA. So really the only thing that's missing here is this one. All the rest on the human and the chimp are almost identical, but that's very close. So if we look up here, the cow and the pig only have one separation as well, so they're very close also. Now, protein comparison, the immunological techniques, the degree of cross-reaction is used to judge relationship. Amino acid sequencing is similar sequence in the same protein indicates a close relationship. Using neutral non-adaptive nucleotide sequences assumes a constant rate of mutation over time. Researchers doing comparative mtDNA sequencing used their data as a molecular clock. They equated about 5.1 nucleic acids differing among the songbird species. So they've seen changes in the mtDNA sequencing from 2.5 million years ago. So they've seen some changes which we are not seeing. But part of that, again, is because we haven't let it change long enough. So again, just looking at a phylogenetic tree, anything close to the trunk of the tree, way over here, is primitive. Anything out to the tips of the tree over here, more advanced. All right, well, that sums up this chapter. 2 more. We are almost done. We'll see you at the next one.